To begin with, the basis of DNA, it stands for deoxyribonucleic acid, three main structures, which is our 5-carbon sugar, which is deoxyribose. We have a phosphate group that's attached to the 5th carbon, or the 5' carbon, and one of our 4 nitrogen bases, which are adenine, thymine, guanine, and cytosine. And you're looking at almost a whole picture of a nucleotide. What you're actually looking at there is a picture of deoxyribose. And you'll notice there is no oxygen on that 2nd carbon. It's just a hydrogen by itself. Let's start with our nitrogen bases. Adenine and guanine are our double rings, which are the purines, and thymine and cytosine are our pyrimidines. Just a good rule of thumb, a purine always pairs with a pyrimidine, and Chargaff gave us all the math for that. And we also know, based on his studies, that adenine goes with thymine and guanine goes with cytosine, or A to T and G to C. These are just the pictures of them. Clearly you can see in this region here it is double ringed, and going over here, let's just use thymine since we use adenine, that is a single ring. So those are just images of the nitrogen bases for you. We've looked at those in the past, but I thought you could use one more peek. Looks kind of chaotic here, but don't freak out. We're just talking about the chemical bonds, and for some reason my heading got all messed up. Obviously, you can see on the imagery that we have 3 hydrogen bonds between C and G, and we have 2 between T and A. We talked about that. We don't treat it any different though. It's still just hydrogen bonds. The phosphate and the sugar bond, however, is covalent. It's very, very strong. Keeps our backbone intact. We don't want that to break, because otherwise we'd have a big disaster. The hydrogen bond, as illustrated with our 2 circles there, much, much weaker. It doesn't affect the strength at all, like I said. Maybe a little bit, but it's negligible. The helicase, once we get the replication, is still going to break it just as easily. And you will have that mix posted very, very shortly right after this one. Nucleotide is just one piece of this puzzle. We've talked about this in terms of monomers and polymers. The nucleotide, though, is just the phosphate, the sugar, which is deoxyribose, and one of the 4 nitrogen bases. That's what a nucleotide is. It's just one little piece of DNA. The nucleotide or nitrogen bases are the monomer. These repeat, which makes a polynucleotide, or just a simple polymer in our case. It's important to know that there is absolutely no pattern to the nitrogen bases. It's completely random, and this all goes back to random selection, law of independent assortment, crossing over. All that stuff sound familiar? Yeah, this is where all that stuff comes into play, especially when we're talking about DNA and how it gets mixed up, which is a big part of adaptations and evolution. Directionality, I've changed the colors for the example down there, which is really, really nice of me. The basic rules of base pairs and then directionality allows us to make any strand of DNA that we would want. Remember, one side is going to go 5' to 3', while the other goes 3' to 5'. We have the phosphate pointing to the 5' direction because that's what it's bonded to, the 5th carbon. And let's just go ahead and make some green hydrogen bonds here. I just made them dots. Close enough. It's not perfect, but it's good enough. When you do this, though, and we'll put this in red because it's important. When we do this tomorrow, do not forget to add your hydrogen bonds. That's a very important concept here. And you can see with our example, we got just the basic ACGT on one side and then TGC on the other. It's kind of a pattern, I think. I was pretty bored doing this, but it's a good illustration of how we can predict one side with the other. Again, though, directionality, super important, especially when we get the replication. I am foreshadowing. That's an English term. Bam, double helix. That's the last major part of this. We twist it up like a spiral staircase. I like this image. It does a good job. It does the different puzzle pieces, complementary base pairs. The only thing it didn't add was directionality, but that's okay. So we can call this one 5' and this one 3', and it's all just the same. And if you follow the strand, which you should, you would know that's 3' there and that's 5' here. Double helix. Let's get back to some historical stuff about it. This is an actual picture of Rosalind Franklin. Remember, she was really groundbreaking. She was a woman doing these things, which was really, really impressive for the time. This was back in the 40s and 50s. But I digress. First off, we have Chargaff. He did all the studies with specifically the nitrogen bases. Rosalind Franklin was the one that determined the helical shape. She didn't get the credit for it, but she did figure out through X-ray diffraction it was the helical shape. Of course, this was after the work of Griffith. Remember, he used the R and S bacterias, R being rough, S being smooth, the smooth having the capsule. And important to note about those Griffith experiments that the reason the mice died is because it was a bacteria that could reproduce with a capsule. Okay, I'm going to say that again. It was a bacteria that could reproduce with a capsule. So when we heat killed the S, it didn't hurt the mouse. But when the mutation occurred, putting the capsule on the rough, then we had a problem. So a S that could reproduce killed, but an R did not. But when the mutation happened and the R formed the capsule and could reproduce, that's when we had dead mice. Dead mice. From the work of Franklin, we got to Watson and Crick with their double helical shape. They get credit for it. They pretty much stole all this stuff. They are not the most attractive human beings on the planet. I kind of told you this when we had the imaging class. What else are they going to do with their time? They're clearly just going to study the shape of DNA. Not pretty people. Don't forget that Hershey and Chase were the ones that did the groundbreaking study that determined DNA was the genetic material. They tagged it with the phosphorus 32 and let the bacteriophage infect the bacteria, and they found all the phosphorus inside of the bacteria, and when they did it with sulfur on the protein, the sulfur was on the outside, and that was sulfur 35, just for reference. So we had phosphorus 32 and we had sulfur 35. Those are just radioactive forms of those elements. And that's it. That's it for this part. This was the history and structure. I'm going to quickly load this up. It's only about 7 minutes and 45 seconds. I'm going to work on your replication one and put it immediate up after this. So stay tuned. You have about, 12 minutes to get the second one.